Wait, just make sure it's.
But it's not the owl. No. You're on the owl.
Oh, yeah. Oh, yeah. Let's just select this one. Oh, that's.
Yeah, that's
Oh,
the power cable.
that's not the owl. Okay, sorry. My God, sorry. Wait, the owl
Okay,
is
there
here.
it is.
Okay, we got this.
I'll work at it.
Okay. Yeah.
No. You can. But I do talk with my hands, you know, I might go like this, knock your computer out.
Okay.
All right.
Everything should be fine.
Let's go under. Hold on, let me make sure, is this on, video's off though?
Oh, no, your
Video's on, okay, no.
let me check.
Can you see it on yours? It's like, yeah. This is one.
But there's no.
So that's so. Because there's no light.
Yeah, I think it's the only piece which is not as bad, but like I checked before, but maybe this one, yeah, not this one. And it's not, okay.
He was saying,
I don't know.
I mean, I teach in this room and we do have the lights on it. I'm just concerned that this is not on. This doesn't sound like it's on.
Yeah, it's not coming out.
Yeah. I don't know. How about now? Yes. Am I louder now?
Yes.
Okay.
Great.
Okay. Do you want to do it in this one? Here you go.
So hello, everyone. Welcome to the last public talk of this academic year, which is the Life on Earth and Beyond panel. It's wonderful to see you all here. Before we begin, I would like to mention that the public talk series are generously supported by the Trottier Family Foundation. And this allows us to run these beautiful events. And I would also like to acknowledge the presence of Lauren Trottier in our audience today. So thanks a lot for your support. And yeah, so before we move on, so today we're going to have Professor Nagissa Mahmoudi from the Department of Earth and Planetary Sciences and Professor Richard Levelet. Thanks so much for joining us. Before we begin, I would like to mention that the Tortoise Space Institute and McGill Physics are committed to equity, diversity, and inclusion. And we want all of you to feel very welcome, because we can only do science when everyone feels comfortable in their own workplace. And so if you ever feel uncomfortable, please reach out to me and I'm happy to support you. If you have any ideas about cool outreach events, you can reach out to the outreach email. We're really happy to receive all your input and your feedback. So this is the code of conduct at our events. So there is some behavior we do not accept. And I'll give you 1 minute to read it. So yeah, so we can start our panel. So today, as I mentioned, we have Nagissa Mahmoudi, whose talk will be The Secret Life of Microbes, How Earth's First Life Forms Conquered the Planet. And then we have a complementary talk by Richard Levelet, How Close Are We to Finding Life in Our Solar System? So I would like to welcome the first speaker, Nagissa. And there's gonna be, again, a switch, so stay tuned. All right.
Okay. Can you guys hear me okay?
Yeah.
Is this on? There's no light on this, so.
There's no speaker.
Yeah. What about now? Yes. Okay, so if I become quieter and quieter,
Here's the second one, I'm preparing it.
okay, just somebody raise your hand. Yeah, so I'm really excited to be here. This is actually the room I taught in this semester. I teach astrobiology on this campus, so I teach about 170 students this semester, and I try to get them excited about looking for life in the universe, how life started on our planet, what do we think the evidence for it is, all of these really interesting questions that a lot of us think about at the Trottier Space Institute. But I'm really excited that this is a public talk. I've never given a public talk here at McGill. So I'm super excited to sort of convey some of these ideas and concepts to all of you and think about the sort of big picture questions about life on Earth. How do we think life on Earth started? Going to give you some evidence for it. What was sort of the evolutionary trajectory, and how is that going to help us understand where we might find life elsewhere in the solar system? So the title of my talk, I have a 4-year-old, so we watch a lot of Secret Life of Pets by Pixar. So this is an homage to that movie, is that Pixar movie where, you know, these people leave and the pets do all of these sort of funny, silly things. And that's how I was thinking of microbes when I was preparing this talk, is we don't really see them, but they're doing all of this invisible sort of reactions and work in the background that's ultimately shaping the habitability of our planet. So my training and sort of what I call myself is I'm a geomicrobiologist, right? So I study microbes in the ocean. And microbes are broadly defined as single-celled organisms that you can't see with your naked eye. So if you look out into the ocean, this is a picture of the Mediterranean Sea I took on a research cruise many, many years ago. You don't see any microbes there. But if you take even, you know, a milliliter, a couple drops of that seawater, you put it under the microscope, all of those blue dots are individual microbial cells that we find in the ocean. So they're there and they're plentiful, we just don't typically see them. So they're really this invisible life form that we're constantly interacting with. So I put this up here to start with because, you know, most people, when I tell them I study microbes, they always assume it's related to disease. You know, I think based on the media and based on our own sort of interactions with microbes in our day-to-day lives, we think about microbes as these agents that cause disease or pathogens. And there are microbes that do this. But I remember when I was an undergrad and I took a microbiology class, and I was shocked to learn that most microbes on Earth actually don't cause human disease. And so myself and other geomicrobiologists, we think about microbes that are not living in the human body. They're not causing COVID, but they're the ones outside in nature doing these other really interesting things. So I call these microbes in the wild, but everywhere we look on Earth, we find these microbes, right? So if you are gardening this time of year and you're planting flowers, there's microbes full, your soil's essentially full of microbes, the grass, the air. And so we find them in these really sort of like, innate boring places, but also these really strange places on Earth. I'm showing a picture here of these really unusual hot springs that exist in Ethiopia. The reason they have these sort of strange colors is that there's this really unique chemistry that goes on in these hot springs. And the pH of it is really, really acidic. It's almost like battery acid. And the temperature is near boiling. And so this is a very inhospitable place to life, but we find microbes living there, thriving and doing really well. So almost everywhere we've looked on Earth, we find them there, and they're really, really abundant. So, I just told you they're abundant, but besides being abundant, microbes are really diverse. When we think about biological diversity of organisms on Earth, and I would ask you to think about that, most of us would probably think about places like coral reefs, the Amazon rainforest, these types of places where we can sort of visually see the diversity and the morphology of different, say, plants or animal species. But microbes are actually the most diverse life forms on the planet. I'm showing you here a very old and sort of outdated but simplified phylogenetic tree. So this just shows you the evolutionary relationships of all the sort of life forms we've discovered. For reference, we're up here, that's us, we're part of the eukaryotes. But all of these other branches here, the bacteria, the archaea, which make up, most of the tree of life, 2/3 of it, those are all single cell microorganisms. So it's really, I tell people it's really a microbial world, and we're just actually living in it. And going back to seawater, so the estimate is that, a single milliliter of seawater, so just a couple drops of it, we could do DNA sequencing and we find thousands of individual species of microbes. If you were to go outside and grab some soil, from just sort of, your garden or anywhere in Montreal, and you were to sequence the microbes that live there, you'd find, as many as 10,000 different species. And so in terms of, beyond just diversity, there was this really interesting paper, and I wanted to share this a couple years ago. And what it tried to do is it tried to quantify how much life exists on Earth based on how much carbon is locked up in that type of life, right? So not surprisingly, the majority of carbon on Earth in terms of life is locked up in plants. That's not entirely surprising. But a huge fraction of it is also found as microorganisms. So bacteria here, which are a domain of microbes, as well as archaea, make up a good sizable portion of all of the biomass that we find on Earth. And so beyond just making up a huge proportion of sort of the diversity of life and the total amount of physical life that we find on this planet, they have been around much longer than any other life forms that you and I are typically familiar with. And that's really what I'm hoping that most of you will get from this talk, is just sort of this temporal or evolutionary trajectory that microorganisms have had. So, if we start here at the origin of the Earth, it started around, 4.6 4.5 billion years ago. I'm not an astronomer or a physicist, so I have no idea if that's, precisely correct. But shortly after that, we have the first evidence for life appearing on Earth from really this abiotic landscape, right? That this planet forms and we get life very shortly after. And this life form not only appears, but it sort of persists and conquers. And so if we sort of continue here, and we still have eukaryotes appearing, which is us, but this is simple eukaryotes. Mammals don't appear until, I don't know, like the last few 100 million years. And so for most of this planet's history, if you were to, I don't know, be an alien and come back to Earth and you came back 2 billion years ago, our planet would have been full of life, but it would have been full of microbial life. And that's really the history of our planet, is that we have been a microbial planet that's been dominated by these single cell microbes. So I mentioned that, around 3.8 billion years, that's sort of our first evidence of when we think life first appeared. 3.8, 3.5, it really depends on the evidence. So what is the earliest accepted evidence of life? It comes from this sort of very simple filaments or carbon-rich filaments that were identified back in the 90s. And so just to sort of, I should mention this, when we think about finding evidence for when life started on Earth, we're very, very limited. We know that it started billions of years ago. And when we go back and look for rocks that are from that time period, there's just not very many. So we're not left with very many options in terms of searching for, when we think the first cells appeared. But back in the 90s, there was this paper that came out that was published by Bill Schaaf, where he described these tiny microfossils that looked almost like a worm, but based on the size, it looked like certain forms of bacteria that we find on Earth now. And these sort of fossils were found in these 3.5 billion year old rocks. And he suggested that these could actually be sort of the earliest fossil evidence of bacteria. And I'm not going to go through a lot of the papers that establish this, but finding sort of evidence for the first life on Earth is almost like finding evidence for life outside of the solar system. It's very controversial and there's always different interpretations. But this paper and this sort of time of 3.5 is now the accepted date we use. So in 2018, there was more sort of subsequent isotopic fingerprinting done on these microfossils, right? So this is one of these cases where, technology changes, right? That paper was published in 2018 and 1993. And by 2018, there's different technologies that we can use to go back and look at those same fossils. And when those fossils were reanalyzed, it sort of confirmed that, yes, those are in fact cells in those 3.5 billion year old rocks. And sorry, I shouldn't say cells, but they're sort of fossilized remnants of very early cells. And so this is our sort of first time point of when we think, or evidence, what we have as evidence as the first life forms is 3.5 billion years ago. But then after that, it becomes a bit easier to identify life, because throughout the rock record at say like 3.3 billion years ago, 3.2, 3, we have these very distinct structures that we're very confident is life, right? A few slides ago, I showed you those microfossils, and it's like, you kind of have to like look at it very carefully, and it could potentially be minerals. But in this case with stromatolites, we're very confident that this is produced by microorganisms. So if you haven't seen stromatolites, you could probably go to a natural history museum. They're very common, but they're these sort of layered sedimentary formations that we know are created by microbes. And so microbes will grow in these sort of slimy mats, and they secrete sort of just regular old slime like they do if you, for example, leave something in the fridge for too long. But in aquatic environments, that sort of slime ends up trapping and binding things like sand and other particles. And over time, you get this layered sedimentary structure. And so this is very, very apparent and present sort of in a lot of those old rocks. So we know for sure by say, 3 billion years ago, or, 3.1, that these existed and there was a lot of them. So we know that there was a lot of life. It wasn't, just like one microfossil that we're sort of debating whether or not it's life. These are some really well-studied stromatolites in Western Australia. These are 3.45 billion years old. There's been a lot of papers published on them, trying to sort of, confirm that they are in fact stromatolites, and it's very accepted that they are. This comes from this region in Western Australia where there is some rocks that are very, very old, but they're preserved enough so we can sort of look and see whether these fossils exist. There's also, and you might be saying, well, where are there stromatolites? Like, I don't see them on the Gulf of St. Lawrence. I've been to the Gulf of Mexico. They don't exist. Where are they? they do still exist. So if you're lucky and you ever get to go to Australia to Shark Bay, there are what we call modern stromatolites. They also exist in the Bahamas. They're not as widespread as we think they were sort of, 3 billion years ago, but you can still find them. And scientists study these sort of modern analog stromatolites to understand how they're formed, so we can get a sense as to sort of what we're looking at when we're looking at these very old fossils. Usually when we find modern stromatolites now, they're typically found in very, very saline places where you have such high salt concentrations that you don't have any animals that are grazers, right? So things that are grazers like eating sort of microbial slime. And so this whole sort of ecosystem would quickly collapse once you have grazers in there. But these two systems, like I said, if you're ever lucky, you're in the area, go check them out. I've never seen them, but they're very well known in geomicrobiology. Okay, so I told you we have evidence for life at around 3.5. We've got these very well characterized stromatolites at around 3.4. And so in that first billion years or so, the first cells were formed. Okay, and so one of the big questions that people are still trying to understand is, how do we think life actually started? And I don't have an answer for you tonight. So if you're coming here hoping for an answer, I can make something up maybe in the question answer period, but I don't have anything definitive, because the answer is we don't know, right? We don't have a time machine. There's theories. I'm going to describe two sort of theories to you, but they're both very much still theories. And depending on who you talk to and sort of what papers have come out, there could be support for either one theory or the other. So the first theory is one that is probably the older of the two theories. You may have heard of this. This was first actually proposed of and thought of by Darwin. This is the primordial soup theory. This is the idea that life started in these shallow ponds on the surface, and that there was these sort of, accumulation of organic molecules. So you had this sort of like prebiotic soup. That's where the sort of soup in the theory comes in. But that you had this sort of abiotic mixture of organic molecules, and then somehow you spontaneously got the generation of life. And this theory sort of really picked up steam in the last few decades because of these very famous experiments that were done. These are the Miller-Urey experiments. So this is just a cartoon of what their contraption looked like in the lab. And what they wanted to test was sort of experimental evidence for the primordial soup theory. So they simulated the conditions of early Earth, and specifically the atmosphere of the early Earth. And then they gave their sort of, gases a spark to sort of simulate lightning. So if you give it a spark of lightning and you have these mixture of gases that we think are present on early Earth, can you even get the organic molecules that you would need to make cells, right? Because this primordial soup theory is contingent on the fact that you end up with these very concentrated mixtures of organic molecules that we know life uses. And so they do these experiments, and then whatever sort of organics are formed sort of come down through here, and then they get collected and they can measure it. And what they found was that they were able to actually generate about 25 different amino acids. And amino acids are the building blocks of proteins. And so this really lent support to this idea that on an early Earth, if we assume that these are the gases that were present, and there are sort of, strikes of lightning, that you could get the formation of building blocks of life. And in theory, those building blocks would then sort of rain down to the surface into these ponds. And if you get enough of these organics, you now have this very concentrated mixture of compounds that we think life now uses. And potentially you could get the formation of these, very early or primitive or prebiotic cells. Oh, something happened. Is it back? Okay. So commercial is what that was. Okay, so yeah, so this idea is that life is starting on the surface of this planet in these ponds. Oh, and it disappeared again on my computer. Okay. Let's see. There we go. Okay. The other theory that I'm going to tell you about is a more recent theory. This was proposed back in 2010. And this theory suggests that life didn't start on the surface of the planet in ponds, but actually in a very different environment. That life actually started in the bottom of the ocean in these hydrothermal vents. So these hydrothermal vents appear in places in the seafloor of the ocean where there's essentially cracks and sort of the crust or the ocean crust is splitting apart. And so you have these very organic rich and mineral rich gases coming up, and they're usually very hot as well. And so when they hit the cold seawater, they usually end up generating, all of these interesting gases. So these are pictures of black smokers. There's also white smokers, but they're very, distinct and unique looking. And oceanographers have documented them, at all of these different mid-ocean ridges, and they've looked at the microbes and the type of life that live there. And so this has been proposed to be a potential place where life started. And the reason is it has to do with the chemistry of these vents. So these vents release essential elements for life. And they also release energy. And the way they release that energy is really in the form of these heat and chemical gradients, depending on the specific vent. So this is a little bit complicated, but it's just to show you that with certain gradients that appear at these vents, you can just have very interesting chemistry occurring. And so they're spewing out this very hot mineral-rich fluids. And we think that could have facilitated the formation of complex molecules that would have made up early cells, or at least molecules that we know that cells are potentially made up of. And so it could have been a potential site where you could have got the origin of life, these primitive cells or these prebiotic cells first appearing. Another reason why this theory is popular is if we think about this, right? So, life starting off in ponds, again, the first evidence is around, 3.5 billion years ago. And I'm not an astronomer, but I'm told that early Earth is a very sort of inhospitable place for life. There's a lot of, you need to go into my computer.
Zoom, the YouTube live stream
Just stopped?
just stopped.
Okay. Do you want
So
me to?
if you could maybe reshare on Zoom, that might be the issue. Yeah, stop sharing.
Uh-oh.
Now it starts.
But it won't do it, so I need to go like this again. I go like this. And then I need to go like that. Zoom. Share. Oh, no. Or it would do desktop too.
Yeah. You did the same before, right?
I did the same before and I don't know why it stopped sharing with Zoom. We try to share directly from the PowerPoint. It's going to do the thing where it shows presenter.
Yeah, scroll down on the, and there's, if you can scroll down, there might be more screen.
There's not.
Yeah,
It
it just
disappeared. There
stopped.
was a desktop too, and now it's completely disappeared.
I mean, it's the YouTube audience.
Number one, I interrupt this. Did you already try just doing the PowerPoint correctly? So just click on the application. Yeah. And then share. And it will just share directly from PowerPoint. Sometimes what happens is that it will do. Let's try it. Let's show what one is in. Yeah. I don't know, can you look at the YouTube? Sorry about this.
I don't know what happened. There was a second screen. It just disappeared.
It was not an error.
In this computer?
Okay, now it's back.
Okay, it's back.
I think if I knew what to plan, I could.
We're working out the kinks for you. So
Yeah.
your talk, no technical difficulties.
That should be fine.
Okay.
Yes, it will.
I won't move too fast. Okay.
Okay, yeah, that's good. Sorry. about
Okay.
that.
All right. So these deep sea hydrothermal vents, okay? Oceanographers have documented them, at all of these different mid-ocean ridges where, these tectonic plates are moving apart and you have this sort of really geothermally heated, hot, organic, rich mineral water coming out. I already went through these slides. Okay, sorry. Okay, this was where I left off. All right. So what is the other reason why this theory has sort of gained traction and why it, makes sense based on our understanding of what the Earth would have been like? So early on in Earth's history, we think that there was a lot of sort of impacts from asteroids and comets and meteorites and that sort of stuff. And so the surface would have been really inhospitable, right? Let's say you're some early primitive microbial cell that's sort of like figured out how to put these organics together. And then, I don't know, like an asteroid slams into your part of the planet. And so deep sea vents are very appealing in that way. In that, your life is not starting at the surface that is, has all this sort of impacts and dynamic changes. It's starting in this very stable environment where you have sort of the stable source of energy, this really warm water, and you're sort of shielded and protected from harmful radiation that we think exists around that time and other sort of impacts that would have impeded the development and the evolution of really early life. So, like I said, I don't have an answer for you, but what I can tell you and what I find very fascinating as a microbiologist and as someone, who teaches astrobiology, when I think about life on this planet, is that it started relatively early after the Earth was formed. So again, it's like, if we're talking about the Earth being formed at around, say, 4.5, and, I showed you this fossil evidence that we accept at around 3.5, it only took about 1 billion years for life to get started and then very quickly sort of, take over and proliferate around the entire surface of the planet. And so this brings us to the million or maybe billion-dollar question that we're always grappling with. And when we think about astrobiology and finding life elsewhere and sort of the origin of life on Earth is life common or is it rare, right? Is Earth very special, did the origin of life maybe only happen once and it's a singular event and we're just really, really lucky? So, the chances of us finding life on Mars or on ocean worlds or maybe some of the places that Rashard's going to talk about, it's not going to happen because this is a very unique and a very sort of rare event. Or is life common? Is this something that's a natural process that when you have sort of certain conditions, organic molecules, reactions, maybe the right temperatures, the right pH, that you're naturally going to get, the formation of these cells and life appearing, and that maybe the universe is full of life. We haven't discovered it, and that this is really not a very special place. that we can find life elsewhere. And also along these lines, this brings up a lot of questions for biologists, is that, we are sort of have evolved from what we think is a single lineage of cells that we call the last universal common ancestor. But is it possible that there were multiple different life forms that existed and all of them went extinct? And we're sort of just, the survivors of the winter, because you could have had multiple origin of life sort of events. And so again, I don't have any answers, but this is sort of, I think, at the sort of basis of when we search for life elsewhere and we think about how life started on Earth. So what I can tell you is when we go to other places and we look for life, or even when we're thinking about how life started on Earth, a lot of these ideas, a lot of these theories are based on what we think are the key ingredients for life. And they can really be broken down into these three broad categories. So obviously water, right? So we think there has to be water around, water involved. The first cells would have evolved in water. Organic compounds. And specifically, we're interested in compounds that we can find that have these 6 elements. So oxygen, hydrogen, carbon, nitrogen, sulfur, and phosphorus. And again, that's because all life on Earth primarily uses organic compounds made up of these 6 elements. And then lastly, a steady energy source. And that energy source could be chemical energy, so something that you could find, say, at a vent. It could be potentially electrical energy. It could be ultraviolet light from the sun and thinking about photosynthesis, but some sort of energy to actually fuel life, right? We agree that there has to be a way that life has to be sort of propelled and fueled. And so again, thinking about places where those ingredients exist, and hopefully, I'm guessing Rashard's going to be talking about Mars specifically and other places. These are places in our solar system where we think those ingredients exist. So, Europa and Enceladus are known as sort of this places in the solar system as ocean worlds, right? So these are places where, it might be covered by ice, but there's a subsurface ocean there, right? And so water is there. Organics are there. There might be some sort of hydrothermal reactions or energy going on there. And then Mars, which, is not overflowing with water now, but we do think once upon a time water was there, and we do have evidence that there are organics there. So now I want to sort of switch gears and think about, I told you that microbes are doing these things that we don't see, that they're really this invisible force on our planet that actually make our planet habitable for us as humans, as eukaryotes, as complex life. And without them, without microbes, sort of doing these reactions billions of years ago and evolving these capabilities, we would have never actually appeared, right? You would have never gotten intelligent life on Earth. So the way that they primarily do that is that they cycle things like carbon, things like nutrients. We're going to talk about oxygen. And they cycle that in a way that makes those chemicals or those nutrients available to us. So oxygen is a very obvious example. Why? Because we've all been sitting in this room for the last, 51 minutes, and we've been breathing it in and breathing it out. And if there wasn't enough oxygen, we would all know really quickly, and we'd want to leave this room. And when I told you about life starting on Earth and, these stromatolites and these 3.5 billion year old, microfossils and rocks, the thing I didn't tell you is that during that time in Earth's history, we don't think that there was any breathable oxygen. So all of that life that existed and appeared and sort of conquered the Earth for the first few billion years did so in the absence of oxygen. It used other chemicals to sort of, run its cells or maintain its reactions. And so for the first, few billion years of Earth's history, there was no oxygen at all. And then sort of in the stage 2, I circled this, it says oxygen produced. And the way that we think that that oxygen was produced was by the evolution and the emergence of these very specific cells and this metabolism that we've now sort of, it's all around us now, that is oxygenic photosynthesis. So this is what our houseplants are doing at home, right? This is photosynthesis where you're taking up CO2, you're taking up water, you're absorbing light, and then you're spitting out, glucose is here, sort of a proxy of organic molecules. But the key thing is here is that you're breathing, these plants are breathing out oxygen. That's what your plants are doing at home. That's what the trees are doing outside. But a few billion years ago, there's these single cell microorganisms that we now call cyanobacteria. So they would have been, ancient cyanobacteria or primitive cyanobacteria, but they developed the capability of doing this, of taking CO2 from the atmosphere, taking up light and doing this reaction. And as a byproduct, just sort of accidentally of their metabolism, because they don't use oxygen as part of their metabolism, but just as a byproduct, they pumped out O2. And so this is how our planet went from being this planet that had no oxygen to being full of oxygen, and oxygen being this thing that all sort of complex life, all intelligent life that we know on Earth uses. So this is just a microscopy image of cyanobacteria. I put this in because you can see cyanobacteria all the time. If you go to any lakes or rivers where it's sort of really warm in the summer and overrun, and you see this sort of green slime, that's usually cyanobacteria overgrowing. So they're everywhere on Earth now. We find them everywhere. They're in the oceans, they're in rivers, they're in streams. They've sort of, dominated the Earth. And so not only do these cyanobacteria evolve and they produce oxygen, but they also allow for the formation of the ozone layer. Again, totally by accident, right? So when you have O2 in the atmosphere, it can react with UV light and you can get the formation of O3, which is ozone. And that's a big deal. Why? Because it really acts as a shield for early life. So now life can expand to regions near the surface of the Earth. Right now, our ozone layer absorbs something like 90% of UVB in all UVC. And so thinking about life expanding and evolving during this time, the formation of that ozone layer would have been a game changer because you're now not exposed to this very harmful radiation that can damage your cells. And so not surprisingly, when we think about the evolution of complex life, and when I say complex life, I mean things that we're familiar with, like plants, animals, fish, corals, all of these sorts of things, it's really closely linked to these increasing levels of oxygen that we've had over the last few billion years. And it all started because of the activity of these cyanobacteria who developed the ability to sort of, take the CO2, take the light, do photosynthesis, but spit out O2. And so, thinking about things like eukaryotes first appearing, land plants, dinosaurs, corals, mammals, all of that was at a time, this sort of blue here is the oxygen curve, but all of those sort of complex life forms that we're sort of familiar with, they all appear when there's enough oxygen around to breathe. And all of those life forms, most of them, besides plants, obviously, usually take up oxygen, right? Mammals are dependent on O2. And so it's very closely linked. These O2 levels are closely linked to sort of the evolutionary trajectory of complex life. And I always put this in because it's such a simple example of how important microbes are. This is an image, a satellite image of sort of the Earth's ocean of chlorophyll concentrations. So these are sort of how we can, figure out where these microbes are. But these single cell microbes exist in the ocean now, and they essentially produce about half of the oxygen that we are breathing in. So yes, plants produce a lot, the Amazon rainforest produces a lot, but the microbes in the ocean now, just sort of by volume and surface area, produce a huge amount of oxygen that sort of fuels all life on this planet. Okay, so the other thing I want to talk about, oxygen is a really obvious example. And the last one I want to talk about is nitrogen. So nitrogen is found in our DNA, and it's also found in proteins. So in order for us and all life forms on Earth to survive, we need nitrogen for our DNA and we need it for our proteins. Here I'm showing you sort of just a chain of amino acids that sort of make up proteins, but you can see the nitrogen is in blue and it sort of forms the base, the backbone of these molecules. And so how do we get this nitrogen, right? The atmosphere is over 70% nitrogen, but the form of nitrogen in the atmosphere is N2. And this is a very sort of inert and unusable form of nitrogen for us. So plants, animals, us, we cannot use N2. We can breathe in N2, but our body cannot use that N2 to make things like DNA and proteins. What we need is quote unquote fixed nitrogen. And so how does life get fixed nitrogen? Or how did early life or complex life or dinosaurs, how did they get their fixed nitrogen? Well, billions of years ago, some microbes evolved enzymes that could take N2, so atmospheric nitrogen, and turn it into a fixed form, typically NH3. And so this process is referred to as biological nitrogen fixation. In the oceans now, we know that those cyanobacteria who are producing O2 are also fixing nitrogen. And on soils, this process is again typically done by these nitrogen fixing bacteria. So different bacteria are doing it on land versus the ocean, but that capability and that sort of nitrogen that is fueling the rest of the ecosystem and the rest of these trophic levels is generated by microorganisms. So some of you may have heard of this, but this idea that plants have these symbiotic relationships with these nitrogen fixing bacteria in their roots that can convert this atmospheric nitrogen into a usable form so that they can grow, right? So without this, plants don't have nitrogen and they can't grow. And about half of the nitrogen in our bodies comes from these nitrogen fixing bacteria. The other half comes from this industrial process that humans have invented to sort of mimic this, to mimic what these microbes do so easily and what they evolved to do billions of years ago. So this is the Haber-Bosch process. This is a process under high temperature and high pressures. I don't know where my mouse is, where you can essentially take N2 and you can convert it into a usable form. And this happens all the time on our planet because this is how we create nitrogen for fertilizers, for crops, for agriculture. And so the Haber-Bosch process that essentially mimics what these nitrogen fixing bacteria, these cyanobacteria are doing has really played a significant role, we think, in increasing our capacity to, feed the human race, reducing famine. It's sort of increased our overall capabilities of what we can do with agriculture. The downside to that is that it has this massive carbon footprint, right? Like these microbes can do it relatively easily. It's like one enzyme, it's relatively straightforward. For us to do it, for us to mimic it, it consumes huge amounts of energy. It's estimated that this process consumes something like 2% of all global energy. So all energy in the world, 2% of it just goes to make fertilizer. And we think it releases about, not we think I should say this, but people have calculated carbon emissions, and this process releases about 3% of all global carbon emissions. So there's a lot of research going in to try to make this reaction, right, that microbes do, but trying to make it industrial, but more efficient in a way that would, release less carbon, but also consume less energy. So I want to just finish here. I have no idea where I am on time. With thinking about, leave you with this idea of where do these microbes live on Earth? And this gets more into sort of the work that I actually do, because I study microbes. I've done a lot of work of microbes in marine sediments, so thinking about them in the bottom of the ocean. I showed you this plot earlier from this paper in 2018 that tried to quantify how much carbon is locked up in these different life forms. And the other thing this paper did is it estimated the fraction of biomass or the fraction of these life forms in these different environments, right? So I want you just to ignore, where's my mouse? I want you to ignore the plants, fungi, the boring stuff, and look at just the bacteria and archaea, right? So these are our microbes here. And so most of them live in the deep subsurface, okay? So the deep subsurface has been, like I said, a place that I have studied for a really long time. And these are environments where really, microbial life is dominant there. A lot of cells are there. And so these would be places like the bottom of the ocean. So, going into marine sediments, going into ocean crust, but also, drilling on land. So, right below our feet, if you drill down, there are tons and tons of active microorganisms there. This is a study from 2015. It's kind of old now, but there's been many since then. This is microbial life. This is a paper published in Science, but showing microbial life that's active 2.5 kilometers below the seafloor. So these samples are collected with these really amazing drilling ships. There was two. There was an American one and there's a Japanese one, but the American one retired. But they have these, drills on the ship where they can drill right down to these very deep depths and get these very pristine, clean samples. And they were able to not only detect microbial cells at these depths, but they were determined that they were active. And so this is really interesting, thinking about how are these microbes making a living there, but also when we think about life elsewhere in the solar system or life elsewhere in the universe, like if we're looking for life and we just go to the surface of a planet and we look around, we're like, okay, nothing's alive there. But even on our planet, life is active 2 kilometers below the seafloor. And so the extent of the biosphere on this planet is huge. And so that tells us that when we do go to these other places to look for life, we really have to sort of, expand our horizons about what kind of life can exist there and how deep it can actually be below the surface. And so microbes that live in these subsurface environments, so either, deep sediments or deep in the continents, they can live under survive under a much larger window of conditions in terms of temperature, pressure, and pH. So things that are, not okay for surface microbes is fine for them. They're actually okay with those kinds of conditions. They have certain adaptations that allow them to thrive there. And so there's a lot of interest in, going to some of these places like ocean worlds and stuff. And yes, they have these very thick, ice crust and that sort of thing. But again, on our planet, that doesn't seem to be an issue. We can go very far down in our planet and we can continue to find active microbial life. So with that, I think I'm going to stop there and allow Richard to sort of pick this up at looking about life in other places in the solar system. So thank you very much for listening.
Thank you so much for the wonderful talk. Just give us a minute to change.
This is the owl.
Testing.
That's the owl. I'll
Okay.
take the mic.
If I may turn it off. And then,
This is mine.
oh my,
Sorry.
sorry.
Me.
Hello.
This one?
Yeah.
We don't
Yeah,
have
it's
the
not-
same issues before.
Better?
But I need to check what is.
Sure. Yeah.
don't know why it's not. It says you're screen sharing, but.
Oh. You wanna try and fix it? So maybe I can slowly get started in terms of to set the scene. So we're gonna leave Earth a little bit. But we're very much gonna remember what we learned about life on Earth. That's very much gonna drive where we're gonna go and what we're gonna look for if we're searching for life. Now, I am a geologist. Nagissa is more of a microbiologist, but she knows some geology and I know some microbiology. Neither of us are astronomers or physicists. That's okay. And I'm gonna focus my presentation on the solar system. We're not going outside the solar system today, unless you have questions about dimethyl sulfide or whatever.
I mean, maybe are you good with the presenter mode on YouTube? That's the question.
On YouTube?
Yeah,
Sure.
if that's
I don't mind. I'm not watching it on YouTube, so.
okay, because then we can just.
Yeah, that's okay.
Okay, makes life easier. Okay, sorry about this.
No problem.
Okay, so I'm just gonna start from the beginning.
Okay, I'm gonna get right into it. So, like Nagissa, we can sort of have these musings about life, and as she very well showed, that life is extremely abundant on Earth, everywhere on Earth, pretty much. And so microbial life is very predominant on Earth today and throughout most of its history, as again Nagissa showed. We also know the building blocks for life are found scattered across the solar system. We talked about those on Earth, and we'll talk about them elsewhere. And there are habitable environments that exist throughout the solar system, either today or in the past. So we can then wonder, well, why is life only found on Earth? And sort of take this approach of what is so special about Earth? I think Nagissa had, Earth is special as one possibility. What is unique or what was unique about Earth, and why did life develop on Earth, and why do we have life on Earth? And conversely, what is missing elsewhere? What was missing elsewhere if there is no life elsewhere? Again, like you, it's okay to not have answers, right? Yeah. So I don't have the answers to all of these, but this can inform sort of our thinking and what we might want to look for and where we might want to go in our solar system or even beyond for that matter. But like I said, we'll stick to our solar system today. Okay, so I'm going to cover one example from the inner solar system, one example from the asteroid belt, and one example from the outer solar system. So we'll look at Mars and the rovers that are working on Mars. We'll look at some samples that have come back from asteroid Bennu, and we'll look at a couple of moons of Saturn, Enceladus, and very briefly Titan if we have time towards the end. And we've already covered this, so I can't do the quiz. You know the answer. We're looking for things like this: liquid water, carbon and other essential elements, and energy sources. And this, again, can inform where we're going to go, what we're going to look for. For life to exist, you maybe want these in some kind of stable environment. And we talked about some possible scenarios for origin of life on Earth. And this would lead to a habitable environment. You have all these things united. Reunited or together. And so, and this is very much what we're looking for: habitable environments on Mars. You'll hear from news from the rovers, or habitable environment or habitable place like Enceladus or Europa. And so, or even habitable planets in the context of exoplanets, which is maybe a little bit different, maybe a little bit more focused on liquid water, although we're at a point now in astronomy where we're detecting compounds in atmospheres around exoplanets, and that's very exciting, but we'll leave that for another time. Okay, this is maybe it looks like a complicated chart. Don't worry about it. It's just showing the relative abundance of elements in our solar system. So on the y-axis is abundance in a log scale. For those who know what that means, it doesn't really matter. And then, of course, atomic number on the horizontal axis. So as you go from left to right, you're going from the light elements to the heavier elements. And of course, in our solar system, hydrogen and helium are the most abundant elements. They make up the sun. That's no big surprise there. But I'll draw your attention to the next most abundant elements, which are oxygen and carbon. Okay, so there's a lot of these elements in our solar system. And what can you form with oxygen and hydrogen? Well, you can form water. Okay, so H2O is actually not rare in our solar system. Now, a lot of it is frozen in many places. And so liquid water is maybe a little less common. And that's very important because frozen water won't get you creatures that are living, or won't get reactions of life to happen without being in liquid form. Okay, water is a good solvent. Those biological reactions require a solvent. And so we're looking for liquid water. So it's very, very important. There's plenty of frozen water, maybe not so much liquid water. There's abundant carbon and all the other elements, the bioessential elements, the Schnapps elements, as we like to call them. Maybe phosphorus is the least abundant of those. And that's sometimes a limiting element even here on Earth. Chemical energy sources are abundant. Nagissa talked a little bit about bacteria that can use chemical energy sources, including at deep sea hydrothermal vents, where they use things like iron and sulfur in different forms and different oxidation reduction reactions, which we don't need to worry about too much. So that's going to guide us now. So we're going to look at these three sort of areas of our solar system, starting with the inner solar system, and of course, planet Mars. And for those of you who don't know, I imagine most people know, Mars is a cold and dry, desolate place today. Okay, it's not very exciting in some ways. There's no liquid water at the surface of Mars. In fact, the temperature and pressure conditions do not allow for liquid water at the surface. And I'm very glad that Nagissa mentioned the subsurface, because that's a whole other realm. Now, so that's Mars today on the right. Mars 4 billion years ago. This is not a picture from any space telescope, of course, but this is what we think it maybe looked like, and it maybe looked like a little bit like Earth, okay? So there's plenty of evidence for liquid water, and I'll show you a little bit of examples. We know that it's lost some of its atmosphere over time, and so it had a denser atmosphere. And so we often talk about a wetter, warmer Mars sometime in the past. This is roughly 4 billion years ago, which incidentally is around the time we're going to talk about when we're going to go look at what the rovers are doing. And that's also around the time when life developed here on Earth, as we just heard. Okay, so it's very, very interesting timing. So Mars has seen planetary-wide changes, environmental changes, climate changes, going from a warmer and wetter planet to a much drier and colder planet today. Okay, Mars Science Laboratory. This is the Curiosity rover. That's the name of the mission, Mars Science Laboratory. It's really a mobile laboratory, including a Canadian instrument that you should all know about if you're paying taxes here in Canada. Curiosity has been going since August of 2012, so that's going on 13 years, if my math is right. It's a well-built American rover that keeps on running. And it's still active today as we speak. And I'll just summarize just a couple of little things. And I'll shout out right away the answer of what Curiosity found. Curiosity found that Gale Crater, where it's been roving for the last 13 years or so, was habitable in the distant past. Okay? How did it find this? Well, there's plenty of evidence for past liquid water inside of Gale Crater, where Curiosity is. These are some rocks that Curiosity has studied. If you're a geologist, you'll be excited by these. If you're not, I'll make you excited about them. These are rocks. These are rocks that were deposited 3.5 to 4 billion years ago. We're not sure exactly, somewhere in that period. And they are rocks that have been, are made of sediments that have been transported and deposited by running water. Not just stagnant water, but running water. And we know this because on the left, that is a conglomerate, a rock made up of pebbles, so fairly large grains that are over a centimeter in size. And that means there was enough energy, the water had enough energy to carry those pebbles, i.e. a river, okay? And on the right is a sandstone, same kind of rock. These are sedimentary rocks created by sediments. Geology is really complicated. A sedimentary rock is made of sediments. Sediments are broken down rocks, little piece of rock, okay? So it's a sandstone on the right, same as the conglomerate, but smaller grains of sand. And, smaller grains of sediment. These are what we call sand-sized grains, and this is a sandstone. And these structures that we see are cross-bedding, and this is a very classic example of a sediment that was transported by water, and we can even tell the current direction if you know how to read these rocks correctly, as geologists do. Justin, can you tell which way the diagram is reversed from the actual image. So beautiful sedimentary rocks inside of Gale Crater. So we had flowing water, i.e. we had streams and rivers flowing at this location 3.5 to 4 billion years ago. All right. We also have evidence for lakes. So I'm skipping ahead a little bit here. So there's other kinds of sedimentary rocks that you would expect to be deposited in lakes. And there's no surprise that at the end of a river, you get a lake. And so that's kind of the setting. Again, this is not, these are not real images. On Gale Crater, the crater was full of water, some people think, or there's at least some water, and it eventually dried out. So on the right is a little simple diagram. You had water in there, you had sediments being deposited over some period of time, you had rivers coming into the crater carrying those sediments. And of course, now that's all gone, and you have a rover that's looking at those water deposited sediments near the lower reaches, but in the higher reaches, you have wind deposited sediments, just like you would expect in a sand dune type environment or a very dry desert-like environment here on Earth. So that stack of sediments inside of the Gale Crater actually records the changes that Mars has experienced going from a wet place to a much drier place. And so that's one of the reasons why Curiosity was sent there, so to be able to examine those changes that have happened. And I won't get into all the details of the different kinds of rocks and different kinds of minerals that are found that are representative of more wet environments and more dry environments, but you can definitely do that, and that's been done. Okay, action shot from Curiosity. So this is a mudstone. So this is a lake sediment, another example of a rock that Curiosity studied. And there it's got its arm out, robotic arm, with the drill, drilling into the, this is the sheepbed mudstone. We like to give rocks names on Mars. And it's actually interesting because, of course, they have code numbers, right? There's hundreds of rocks that Curiosity has studied. And there are code numbers if you want them. But nobody remembers rock 603-38, right? But you remember the sheepbed mudstone because that was a really interesting one. And we drilled into it. And on the right, that's a little bit complicated, but that's from the SAM instrument, where they take a sample of rock, the powdered sample, and they put it into the instrument. And one of the things they can do is they can heat it up. So this is temperature going up to 800 or so, 900 degrees. So they heat up the sample, and then gases are given off, depending on what's in the starting material. And so some of these curves here represent on the right there, or in the middle column, if you will, these are the volatile compounds that are given off. And I don't even know the names of all these, you don't need to know them, but a lot of these are organic, simple organic compounds, ones with carbon, hydrogen, and sulfur in this case. And they're relatively simple. They have smaller number of carbons, for those who know a bit of organic chemistry, 4, 5, 8 carbons. Okay, these are simple organic compounds, but they've been measured in this 3.5 to 4 billion year old rock. So that's exciting, right? Okay. This has been known for some years. This is a paper from a few years ago, and just recently, in fact, just a couple of weeks ago, a new study from the SAM team doing some different manipulation, plus doing a lot of work back on Earth to understand the data that they're getting from Mars. They announced that they found more complex or longer chain organic compounds, and this is just a little NASA promo shot. So the organic compounds in this case have 10, 11, and 12 carbons. So decane, undecane, and dodecane. And I can say that because they're written on the screen. Okay. So a little bit more complex, a little bit longer chains. Now these are Still fairly simple organic compounds. So, you know, we're not talking finding life. And in fact, Curiosity is not designed to search for life, just so you know, just so you're not disappointed. In case you're like, man, it's been 12 years, almost 13 years, and they haven't found life yet. Curiosity is actually that's not part of the mission objectives. It's searching for habitable environments. That's what it's doing. And mission success, it's already shown that Yale Crater was habitable in the past. Okay, so the organic compounds, they could be of different origin. Number one, they could be high temperature, formed by high temperatures in volcanic or igneous processes. They could be from interplanetary dust particles, what we'd call exogenous, so they come from elsewhere. We know that these kinds of compounds exist. Or they could be biological. That's, you know, one hypothesis or one possibility. Unfortunately, with Curiosity, there's absolutely no way of telling apart these possible sources. Okay, so we cannot tell if they are formed biologically or there's some other way that they're formed. However, the fact that we can measure these on Mars in a very ancient rock is very promising for future studies, future missions, or sample return missions. Because actually, the SAM instrument is not really good at measuring complex molecules. Wasn't really designed to do that. So if there is a nucleic acid or something kicking around, we might want to bring those samples back to be able to measure that here on Earth. And so I'll talk more about Mars sample return as we go. Well, actually, speaking of Mars sample return, this is the first step in Mars sample return. So this is the Mars 2020 mission, the other NASA rover mission. This is the Perseverance rover, and they kind of look alike. If you want to know how to tell them apart, look at the wheels. Okay, the wheels on Curiosity are falling apart. They were poorly designed by the engineers, and for Perseverance, they improved the wheels. Okay, and there's some other differences as well, but that's the most obvious one. Perseverance, if you look at the rock in the foreground, there's two holes in the rock. It's also drilling in the rock, but rather than collecting powders like Curiosity, it collects little cores of rock, about the size of a finger or so. It collects those and it puts them in a little container for future return to Earth. That's actually part of the objective of the Mars 2020 mission, to collect samples for future return to Earth. Okay? There's only so much we can do with a rover on Mars, even though these are $2.5 billion rovers or whatever they cost, with amazing instruments that were developed over years. The instruments are rather simple. They're miniaturized. They run on very low power. They're nothing like the instruments that we have in laboratories, you know, here, maybe not in this building, but you know, around campus. And so there's a strong push to get samples back to Earth. And that general idea is called Mars sample return. And we'll say a few more words about that. Perseverance also Perseverance has been on Mars for about 4 years, a little over 4 years now. It was a pandemic rover, almost didn't launch, but they got it done in time. And it's exploring Jezero Crater. I don't do I have that anywhere, maybe not here, but so this is inside of Jezero Crater. For some, it might look like same crater as the other place, a bunch of dusty rocks. But these rocks actually are very different. This was a bit surprising to the science team. These are not sedimentary rocks, but these are igneous rocks. So a lot of the crater rocks are volcanic or igneous, and you know there's reasons for that and explanations for that. But nevertheless, we still see that these rocks have been altered by water. So yes, there was a lake at some point. These volcanic rocks are younger, but there's still some indication of a reaction with water. So we see minerals like carbonates, sulfates, silica, iron oxide. These are rocks that are minerals that form due to weathering of water interacting with rocks. And then elsewhere, we did find indeed sedimentary rocks. So this is a close-up of Perseverance now drilling into a rock for those who like drilling videos. And in this particular rock, sedimentary rock, they found a lot of silicon carbonates, which are good minerals to preserve organic matter or to make fossils. And some of the earliest fossils of life were found in silica, the apex chert that Nagissa showed. And so this is exciting. Now, again, Perseverance is limited in what it can do, and it's probably not going to find those little, remember the picture that Nagissa showed, those little tiny organisms, some of the oldest fossils of life on Earth? Do you think a rover would be able to detect those on a planet, I don't know how many tens of millions of kilometers away? So that's the challenge, okay? Even on Earth, the challenge of showing that there's life in a very old rock is very difficult. And I can guarantee you those samples from, Western Australia or, the stromatolites or the apex cherts, they've been analyzed by tens of people in different laboratories using all kinds of different instruments, including analyzing them with new instruments as they become available, or old instruments that are improved. And we just can't do that level of analysis with rovers on Mars. More recently, this weird rock, this might not mean much to many of you, even the geologists in the room. This is a weird rock, but it's exciting because it's got these, it's got a number of things. It's got, sulfate veins, the lighter tone material. The reddish material is iron oxide, so that's exciting. We like iron oxides, probably formed by interaction with water. And then there's these weird spots which have sort of light areas and dark areas, which we think are redox gradients, oxidation reduction gradients. So going from, say, oxidizing to reducing conditions. And bacteria really like gradients, as we also saw earlier. So this is exciting. And people have shown that this is the kind of stuff that microbes can do here on Earth. And also the Sherlock instrument on Perseverance found organic matter in these rocks. So this has been called a potential biosignature. Now, what is a potential biosignature? A biosignature is a sign of life. A potential biosignature it could be a sign of life, but we're not sure. We need more information, more data, i.e., let's bring those samples back to Earth. So this is another example of a sample that's been collected. I think I'm going to skip this. And in case you're wondering, these are the kinds of things we look for when we're looking for biosignatures. There's all kinds of things at different scales. Yeah, it just gets really complicated. That's part of the problem, though. Searching for life, there's no single easy thing we can search for. It's complicated, even for life on Earth when we're going back 3.5 billion years or more. Okay, so this is the idea of Mars sample return. It's actually being worked on jointly between NASA and ESA, or at least it has been until recently. Who knows what's going on with NASA these days? But and so the idea is that first of all, it's going to be a complicated mission. We should maybe partner up with people and try and figure this out, and also try and pay for this. So signs of microbial life may be only discovered by instruments or by humans using a combination of instruments and techniques here on Earth. Okay, so the chances of finding microbial life that's 3.5 or 4 billion years old preserved in rocks on Mars is unlikely with just a rover, even if it's like a super duper rover. So the proof that life exists on Mars or anywhere else for that matter probably needs to be extraordinary, and that's difficult to do with a rover with a few instruments that can take a few measurements. You probably really need to analyze those samples in quite detail. And there's a good example of that from the study of the Martian meteorite. Those of you who know ALH 84001. So this is a meteorite from Mars that fell on Earth it was discovered in 1984. And that rock had been studied by a number of scientists. And then in 1996, there's a paper that was published. It got a lot of news, including President Clinton at the time made a press conference. And they claimed that there is evidence for life from Mars in that meteorite. And obviously bold claims. And then that rock became probably the most studied rock on the planet. People analyzed it, reanalyzed it, looked at it different ways, different instruments, et cetera, et cetera, et cetera. And most people nowadays claim or believe that there was either contamination from Earth, other explanations for what they observed, or just not microbial life. But it's hard. It's hard to show that yes, there's life in a preserved in a rock, or not preserved. So this is Mars sample return, the scenario, and that's not a typo. There's a study recently that evaluated the cost of doing this over $8 billion US, and that's pre-current administration. So who knows what's happening with Mars sample return? Probably not going to happen anytime soon with the current administration. But the idea is, so there's Perseverance. It has collected its samples. You send a spacecraft to Mars that can launch an ascent vehicle. You have to have a orbiter that's going to catch the ascent vehicle, and you have to go get the samples that Perseverance dropped. So Perseverance has dropped some samples. It is also carrying some samples, and you can either bring Perseverance to the lander or the spacecraft, or you could go get them with some other smaller rover maybe, or more recently, they're looking at using a drone-like spacecraft because of Ingenuity. Does everyone know Ingenuity? The little drone that was piggybacked on top of Perseverance as a tech demonstration. Turns out it worked really well, so we can fly drones on Mars now. And it works so well that engineers are saying, well, maybe we can use the drone or a drone to go get those samples and bring them back. And so that's being looked at and considered now. And then you have a little rocket in this launch pad or this launch spacecraft, and you launch the package once you've collected all the samples. A spacecraft catches it in orbit and then turns around and returns to Earth. No sweat, right? We've never done any of these things. We've never launched anything from another planet. We've never caught anything around another planet. So this is a big deal. But the scientific drive for getting these samples back is so good. And it's not only the question of life, there's a number of other questions we can answer with these samples, like determining the exact ages of these samples using radiometric dating techniques that we can't easily do with a rover. So there's a strong desire to get those samples back. Okay, I think we're probably, we should move on. Okay, Mars, plenty of evidence that there's habitable conditions in the past, there's organic matter, et cetera, et cetera, et cetera. But maybe to actually find signs of life or biosignatures, we might need to get those samples back. And speaking of returning samples, we're gonna go off to the asteroid belt now. And this is asteroid Bennu, which was visited by the OSIRIS-REx spacecraft a couple of years ago now or so. These samples were returned to Earth in 2023. So there's a nice 3D image of the asteroid. It's a rubble pile asteroid. It's not a big chunk of rock. It's a bunch of loose rocks held together loosely by gravity. And there's a sampling vacuum head that was not really a vacuum. That's the sampling device as it was just about to enter the surface of Bennu. And I say enter the surface because it actually kind of entered the surface and there's a big puff of dust. And in fact, so much that the sample container was full and they had trouble closing it. So they got too much sample at first. They managed to close it, get the sample back. And here's the sample container on the left in the Utah desert where it landed. And then finally in Houston at the Johnson Space Center where they opened it. And those are pieces of asteroid Bennu. They got about 60 grams of sample, and they've, of course, been studying that, and they found a bunch of wonderful things. So the samples from Bennu are rich in volatiles with more carbon, nitrogen, and ammonia than samples from another asteroid return mission, the Japanese Hayabusa 2 mission, which went to another asteroid called Ryugu, and much more than most meteorites. So this is a carbon-rich asteroid. That's partly why it's chosen. So they found lots of compounds, including 33 amino acids, of which 14 are used by biology here on Earth. Biology uses a set of about 20 amino acids. They found 14 of those, as well as a bunch of other things that we don't need to worry about. Nucleobases, they were on Nagissa slides, so we can mention those. All kinds of other compounds, and all kinds of interesting salts, including carbonates, sulfates, and phosphates, which were formed by evaporation. So Bennu's parent body, where it comes from, was water-rich. And you had potential for chemical reactions with these building blocks of life and potential for prebiotic chemistry. Now, it doesn't mean life, okay? So these results do not show life. And by the way, these samples are still being studied, including 4% of which will be available to Canadian scientists because Canada contributed an instrument to the OSIRIS-REx mission called the OLA instrument. And because of that, we get 4% of the sample being returned to, sent to Canada for Canadian scientists to access more easily. So these results do not suggest life, but they do suggest that the conditions necessary for the emergence of life were likely widespread across the early solar system. This, of course, increases the odds that life could have formed on other planets. Could have. Increases the odds. Now, are you a betting person or not? Who knows, right? So no life yet, but very interesting results. And then finally, we'll end with the outer solar system. Enceladus, beautiful moon of Saturn. Relatively small moon, but it's a ice-covered moon, one of the brightest objects in our solar system. And it's got these geyser-like jets spewing material out from them. This is from the Cassini mission a few years ago. Enceladus has an ice-covered crust with probably a liquid water ocean underneath, with maybe some hydrothermal vents down at the bottom of the ocean. And we know the importance of hydrothermal vents. And the stuff that's coming out includes ice grains, so water ice grains with a bunch of chemistry, various compounds, sodium, potassium, et cetera, and then more recently, they reanalyzed the data and found evidence for phosphate, so sort of the missing phosphorus in this equation. So Enceladus is habitable today. It's got liquid water, it's got organic compounds, carbon compounds, and this is going on today. We need to go back. We need to go back and not just orbit Saturn like Cassini did. You got to get in there somehow. And in fact, this is one of the priorities. If you know the Planetary Science Decadal Survey, where they give priorities, an Enceladus mission is one of the priorities, but not yet on the books, as we say. And then finally, I'll just plug this mission. This will be one of the coolest missions ever. The Dragonfly mission, going to Titan, the largest moon of Saturn. Titan is a very interesting moon. Do I have pictures? No, I don't have pictures of it. It's got a dense atmosphere, and it's got lakes and rivers on the surface. Not of liquid water, though. Of liquid hydrocarbons, methane and ethane. So it's about minus 160 degrees on the surface of Titan. But there is a methane cycle, just like the water cycle here on Earth. So in some ways, maybe life is not found at the surface of Titan, but there's maybe also a subsurface ocean. But in some ways, the chemistry on Titan resembles some of the chemistry on early Earth. And that reducing atmosphere without oxygen that gave rise to life somehow on Earth, well, this is maybe another example of a laboratory, an analogous laboratory where we can study some of those conditions. So that will be launching in a couple of years, 2027, 2028, arriving 2034. You gotta be patient with these missions. They take some time. Okay, so just maybe to end, the building blocks for life are not rare in our solar system. Habitable conditions, environments are not or were not rare in our solar system, depending, today in some places, in the past in other places. So life must be out there, right? Well, finding signs of this life may require well-chosen samples that are likely returned to Earth from our careful study. So to answer the original question, we're going to have to be patient. I also don't have the answer. But it's exciting. And so we'll end with a sunset on Mars.
So much for the amazing talks. So this went a little bit longer than expected, but we are having a short Q&A session where the two speakers can interact and answer all the questions. So yeah, this is gonna from you. And we also have microphones. And I asked you to repeat the question.
Okay. Okay.
But I think
Careful, because if you pull us taking it out of there.
Who understands the physics of tangled cables? No? Yes, it's on, okay. All right.
yeah,
I think we're supposed to sit down on these seats.
if you want.
Yeah, I don't know, I'm okay standing. You're okay standing? Okay, shoot, fire away. Yes, we'll start off here.
Yeah, Yeah, so excuse it if my question isn't really that refined. McGill credentials have been active for like 20 years, but anyway. So, can you say more about like how the nature, like the dynamics of like the superfluid core, like particularly neutron star, can influence like planetoids with the planets that surround it? Like, let's say you did have a telescope that could get data, you know, about at these planetoids, maybe you could analyze the neutron star itself to learn more about what that are. Like, what data?
It's beyond my expertise. Yeah,
Like,
Yeah. I'm sure there's. What's that, sorry?
I just wanted to feel like.
Well, I mean, maybe say why, where it's coming from, that question.
So I'm only firing this one because I was just interested in like, I feel like it would be easier to lobby for like with respect to government, the prime minister to give you funding to like create interesting types of life using black hole technology, like, you know, creating even more from that versus just like trying to like look for life.
Okay, well, first of all, knowing quite well, you know, the Canadian Space Agency, over the years, they've tried hard to lobby government, including various prime ministers, including various ministers, including some who were former astronauts. And once in a while, some people in Ottawa get excited, but it doesn't come through with budgets. You know, the reality is such that, you know, money is limited. And I don't want to get too political, but I mean, we can't do everything. And you know, the Canadian Space Agency and the space community do a lot with what they have.
So
are we as
a university lobbying right now for like our space related efforts, like actively?
Well, some parts of the university. I mean, we have you know, our director of the Planetary Space Institute and members that are present as well. And you know, various faculty, but I don't think, I'm not sure how much of a university priority it is. Just like it's maybe not a government priority. So whether it's searching for life on Mars or studying black holes, you know, there's, yeah, it's interesting, it's science and innovation and so on, but none of that seems to get people in Ottawa, or certainly not people with the purse strings, all that excited. Now, sure, go ahead, Nikki.
I did not get a question.
I had a question.
Yeah, please continue now.
Okay, well, that's all I'll say there, I guess, yeah. Maybe not a great answer.
I have the next question about like the projects. If like you contaminate the fund, like does the agency like gets, is it accountable for it?
Okay, so good question with a big, long, broad answer. So there are questions of what we call in the lingo planetary protection, which includes protecting other planets and includes protecting Earth. And I like to joke, you know, I ask people, do you think there's life on Mars? Or people ask me, and I say, yes, there is life on Mars. We've brought it there. Okay, so the spacecraft that have gone to Mars, even though we try to clean them as much as possible, and in the old days, like in the Viking days 50 years ago, we talked about sterilization. We know now, we were naive back then, we know, like, you know, smart microbiologists know that that's impossible to sterilize a complex spacecraft with, you know, computer boards and parts and so on. And so we try to reduce the bio load, is what they say. There's different ways of doing that. So yes, you don't want to contaminate another place, and you want to minimize that.
But we've probably already done it.
Yes.
I think it's accepted that we've contaminated Mars.
Yeah.
But in terms of Mars sample return coming back, so we don't know when the samples are coming back, but there are people currently at NASA already working on how those samples will get sterilized in a way where if there is life there, that it could be a threat to us. So those samples will be sterilized in a way that will reduce, essentially, we don't want to destroy the evidence for life while we sterilize it. So this is something that NASA is like actively working on, and when those samples do come, there's a whole procedure that will happen as to where those samples will go, how they will get handled, and they will get sterilized before they're sent out to these labs and these fancy equipments. Yeah.
And again, it's not my area nor yours, but
No.
there are experts on campus here in space law.
Yeah.
I don't know if there's anyone in the room here that can maybe say a few words about that, but Yes, this is a real thing at the international level. Yeah.
You both talked about the necessary ingredients for life,
Yeah.
water being crucial to it.
Yeah.
What about other media for life to possibly?
Beer.
Here, I know. So you don't need water, right? So water, life on Earth uses water, and water is an amazing solvent, so it can dissolve all sorts of different things. And so on Earth, when we think about the liquids that would have been around, water is advantageous for chemical reasons that I'm not gonna get into. Titan is very exciting to us as microbiologists because you have liquid, but it's hydrocarbon chains. And so it could function like water, right? Because what does water do in cells? It's just a way of moving things in and out of the cells, right? It does it at certain temperatures. We know that, it's well characterized. But you could have, in theory, life on Titan where the biochemistry is completely different because that media, right, those hydrocarbon oceans or whatever we think is there, it could still function in the same way. So it's definitely possible. It's not like something we, discount, but this gets into, Richard had on one of his slides, life as we don't know it. How do you look for life if we have no idea what it looks like? So we know what the biochemistry of life looks like when it's using water and DNA and proteins and RNA. I have no idea what the biochemistry is like for life when it in theory can use hydrocarbons as liquid media. And so this is one of the other challenges is not only going to these places, but there is so much debate even among people that study microbes about what is a biosignature. What is a good biosignature? What are we looking for? Like, it just becomes really hard to look for life if you have no clue what you're looking for.
And there's there are experts looking at what are known as agnostic biosignatures. What would any life look like?
Yeah.
Different from ours. And for example, one area of research is complexity of molecules. I mentioned and Nagissa talked about building blocks, simple molecules that are found on Mars and asteroids, and I didn't talk about comets, but elsewhere. These are simple molecules that can be formed by chemical processes. Now, at the other end, you get more complex things like DNA, let's say. No one knows how to form DNA in a simple chemical reaction. There's multiple steps that are involved, and people are looking at complexity of molecules in terms of how many steps do you need to do to put the different parts together to make that molecule. And life is very good at making those multi-step molecules. But so if we find some weird molecule somewhere and we sort of work out their chemistry and how they were formed, maybe that could be a sign of life. But it's a big maybe, and then how much are we able to understand of that life if we don't even know what it looked like to begin with?
So just since you were talking about that we cannot completely sterilize a spacecraft when you're sending it over to Mars, is it feasible or is it possible, ethically feasible, to try to propagate earthly life on another planet by sending it over in a very controlled and closed medium and try to see what happens instead of just like searching for life?
So that would currently be against planetary protection regulations, I believe. Now whether someone would do that, I don't know. Yeah, that would be sort of a no, and you wouldn't be liked by a lot of people.
People have done it with lunar samples.
Yes,
though.
yeah.
So people have taken Martian samples are like gold, right? But lunar samples are kind of like, I don't want to say a dime a dozen, but they're more common. And people have taken lunar samples and they have tried to grow plants, they've tried to grow cyanobacteria, they've actually tried to grow life and said okay, if you take a lunar sample, does it have nutrients to support life? So people have done this. You could do this in theory with Martian samples, but I think it's more of an ethical question.
Yeah.
Do we want to seed our life there on purpose versus okay, we did it with the Viking missions by accident? Yeah.
Or sorry, to repeat the question.
Okay. Well, I think last, I don't know. Do you have a question? Yeah.
Well, maybe more a comment. You mentioned that we know their basic elements are common on planets, maybe in the universe. However, you mentioned that the process on Earth here, for example, for the microbes to create oxygen was accidental.
Yeah.
Although was accidental.
Yeah.
So it seems to me that it is going to be pretty rare to have life, let's say evolved life, as we know it, even oxygen based, because that's the only life we can speak about, is the one we know about. I don't know if there's any other life, so I would be more inclined to say that life is very rare, and my point is, even if we find that there's a lot of basic elements, I mean, what's the point? I mean, are we getting any closer to resolve this issue? I don't see really the purpose of this. Maybe it's just a comment.
Well, no, it's a valid comment. You know, when we send spacecraft to Mars or elsewhere, looking for life is usually not a thing, actually. We've only looked for life really as a mission objective with the Viking mission in the 70s. That mission stated goal was to search for life on Mars. Since then, because we're less naive and we know more about, you know, a lot of things, NASA, for instance, has taken a stepwise approach to first look for signs of water, and they did that with past missions like Spirit and Opportunity rovers, and then they started looking for habitable environments with Curiosity, and then only more recently, you know, collecting samples, and the stated objective with Perseverance is to search for biosignatures. Not necessarily life, but, you know, maybe signs of life. And because we know it's very difficult, but at the same time, we're also doing many other things. For instance, Perseverance has an instrument that made oxygen from CO2. You know, for future humans, we're going to need to breathe on Mars. You know, can we do that? Yes, we know how to do that. We can produce oxygen from CO2. So these are complex missions with many objectives, and searching for life is, while it, you know, catches the headlines, and every, you know, month or so there's a new study that maybe life is found either, you know, in our solar system or around an exoplanet. That's partly the media, right, that likes to get excited about these things and sort of extrapolate or exaggerate claims. But we're doing lots. We're exploring our neighborhood, whether it's our solar system or, you know, exoplanets around us. And I think that's part of who we are as humans, to explore.
That's fine.
Yeah. And if we don't find life, you know, that's a result, right? Like, why is there no life elsewhere? If all these ingredients are there, and the conditions are there or were there, you know, depending on where you're looking.
The last comment on this.
Is it just a rare random event?
Maybe not, but you know, I'm thinking about the Fermi paradox.
Yeah.
Because of
Right.
the size of the universe,
Yeah.
even if there's intelligent life somewhere, what are the chances that we're going to be able to communicate? Nil. That's the distance.
I don't know. Is it nil or is it small?
Well,
You know, we could
that's,
debate
you know,
that, right? Yeah.
Because of the size, the speed of light.
Sure.
time
Yeah.
it takes
There are
to
realities.
communicate.
Yes. Yes.
It's going to
Yeah.
be, maybe there are life, but, you know.
Yes.
Are we ever going to communicate?
Yes. Over here.
The basis of life is evolution, which sort of presupposes that there was one event.
Yeah.
Is there, you know, that we're looking for life on another thing, is there any evidence or possibility or investigation that there was another event that percolated along for a while and then transpired or expired, given the changing conditions of the earth? The suggestion is that maybe there was more than one nucleus.
Yeah.
And one
Yeah.
evolution. I have no idea what I'm talking about, but.
No, it's something that comes up a lot, right? So if life started, did it happen once and we're survivors of that one chance of it? Or did it happen multiple times and we're, you know, like somewhat extinct and, you know, we haven't detected it? So there's different ways to think about it. So the idea that all life on Earth came from one universal common ancestor, that's very strong. There's very strong evidence about how all of us use what we call like the same molecular code, the same DNA evidence that exists. For many years, people investigated this idea of a shadow biosphere, which is exactly what you're talking about. That there was this other life form, microscopic, that started and it's living all around us, but we're just not detecting it because we're looking for, normal DNA-based life. And there was arguments that there's a shadow biosphere that's using different elements like arsenic. A lot of that work's been debunked. In terms of whether life started multiple times, we just don't know. I mean, I've even talked to, very well-known astrobiologists about this, and they said, yeah, it's totally possible it started at the surface and also in deep sea vents. And then the surface stuff probably went extinct because there's all these impacts and all of these changing conditions. And then we're sort of survivors of, what was in the vent. So it's a big question mark. We just don't know. If it did start multiple times, that would be fascinating because now we're talking not only about life starting in a short amount of time after the appearance of Earth, but it happening multiple times. And then it's no longer rare. It's actually maybe really common. Yeah.
You guys keep on saying like things that relate to Earth, but why are people fixated on what is not on Earth other than thinking about stuff that actually happens elsewhere with a different information instead? So like there's like bias towards Earth and life on Earth. Like what is
So the problem with the people looking or the people doing the work is your people. Humans have biases. And I've seen this multiple times, working with a 400-person science team with, Curiosity or Perseverance. You get a picture from the rover one day and it's a blue rock on Mars. And then everyone starts emailing their favorite blue rock from Earth that they've seen in some weird field site. And then, the rover takes another picture at a different illumination angle or a different time of day, and the rock is just gray. And then, the emails stop and people aren't excited anymore. Or, the first rock that Curiosity studied, the first big rock that I encountered, got out all the instruments, took, microscopic images, chemical analyses, the whole deal. And then half the team thought it was a sedimentary rock and half the team thought it was an igneous rock. And I didn't do a study, no one studied this, but I bet those who thought it was a sedimentary rock were people who studied sedimentary rocks, and those who thought it was an igneous rock were people who studied igneous rocks. So humans are biased first of all. Second of all, we have n is equal to 1 in terms of life, right? It's the only example we have. If we had a bunch of different life, well, sure, it's a bias, but what do you look for? That's what Nagissa was saying before. What do you look for if you don't know what it looks like? Right.
I just say that other things that not be.
Exactly. And I wouldn't say people are fixated with Earth, but it certainly informs us of maybe the easiest thing to look for and maybe the most probable thing, perhaps. But people are aware of this bias. And we gave some examples before of some people are thinking or considering what else could we look for? How would we look for something that we don't know? Do we look at people have looked at electrical sort of electron transfer processes? Can we detect those? I talked about the complexity of molecules, chirality of molecules, and life on Earth has chosen one, right-handedness or left-handedness of molecules. If we go, the molecules on menu, it's kind of a mix of left and right. And some people were disappointed by that. They were hoping maybe if they found all one-sided handedness, that would be what life used to start on Earth. But I just think that shows that life has selected that on Earth. But some people are not entirely fixated or entirely biased, but there's some bias. And it's just hard, But there are smart people trying to think of.
But I think that there's assumptions that we can make just based on chemistry and physics and just biology of life. So for example, Richard put up pictures of meteorites. Meteorites are full of organic molecules, right? They're full of amino acids, organic molecules. Organic molecules by definition are carbon and hydrogen based molecules. So we think it's likely that life is going to probably use or be composed of organic molecules. Okay, so if that's an assumption we make, we're probably not going to go to lava worlds because we know about the thermal stability of organic compounds. So yes, like we do use Earth as sort of our proxy because we do only have an n=1, but I think there are some boundaries that we can make based on our understanding of not only what chemistry life uses, but also what sort of chemistry we find and compounds we find in, lunar samples or meteorites or this or that. So that sort of stops us from, we're not going inside of like, the surface of Venus. That's not a place that we think life exists. I think it's like 400 degrees Celsius or something like that. So, carbon bonds would just break up, right? So we don't think that there's some sort of life there that's like not using carbon as figured out how to live on the surface of Venus, for example. Do you want to do the online question?
Yeah. And just quickly, people do invoke sort of non-carbon based life forms. Like what about silicon? Because silicon can make a lot of bonds with oxygen and so on and other elements, but it's not 400 degrees, and it's we don't know a way of doing that with producing genetic code bearing information or some kind of genetic information and so on in complex biochemistry. It's pretty much an X-Files episode for those of you who remember that one. Right?
I think we're gonna stop with the last question by our online audience,
Okay.
which is, so Nagissa, you mentioned stromatolites in your talk, and the question is, was there evidence of stromatolites on Mars?
Oh, that's controversial. I'm gonna let you
No. I would have started with that, but yeah, by thought. But no evidence of stromatolites. There have been some fairly serious analyses of some of the rocks, the images of the rocks. People who sort of see what are known as biosedimentary structures, not necessarily stromatolites, but things that on Earth can be somewhat related to microbes, but there's not a lot of convincing evidence for that, and most people don't believe that. And certainly not any obvious stromatolites. That would be the mission chief scientist for the Mars Science Laboratory is a stromatolite expert, or at least the previous mission scientist. He passed the torch on that. No stromatolites. But the rocks, just to remind you, the rocks that Curiosity and Perseverance are examining, they're not the exact same age, but they're 3.5 billion to 4 billion years old. And these are rocks that on Earth are very rare because they haven't been well preserved. And so finding evidence for ancient, earliest life on Earth is pretty hard to do because it's hard to find the right rocks that have not been totally transformed by plate tectonics and dynamic processes here on Earth. But on Mars, all those rocks are nicely exposed. So we can more easily study them in some ways. And we have not found stromatolites.
Thank you so much for the panel. Thank you for being here. And I wanted to thank also the audience. So this was the last talk of the academic calendar. So summer, yeah. And I'm gonna see you in September, or yeah, we'll be here with a new public talk. And I wanted to again mention that we really value the support by the Trottier Family Foundation, because it really made this possible. And you also made this possible. So thank you. Sorry for
No, it's nice to try and make work.
Yeah.
I never did it during the pandemic, but I went on, even when I came.
Oh, yeah, yeah.
I have no idea.
But even people who have done it all through the pandemic, it's still like just. Maybe.
It's like random stuff. Okay, that's garbage out.
So this we can stop?
Yeah. Yeah. You can play. Yeah, you can. Yeah. Here, and you do your.